So welcome to video 2 of Biology Paper 1. So this is looking at Unit 2, focusing on organisation. So if we start just with the basic principles of organisation first of all, so when we're thinking about the organisation within a living organism, we start off with cells, which are the smallest building blocks of a living organism. They obviously contain smaller things such as our subcellular structures that we looked at in video 1, and those subcellular structures eventually, if we come down to it, are made up of atoms. So we start with our cells. Group of cells working together to carry out a function is called a tissue. Group of tissues working together to carry out a function is an organ. Organs working together to carry out a function would be an organ system, so things like the digestive system, the nervous system, the immune system, and then those organ systems collectively make up that whole organism, which, for example, in our case, could be humans. So you need to know that order kind of from smallest to biggest and be able to describe what a tissue is, what an organ is, and so on. So moving on then to look at our first example of a system that we need to know, so an organ system, we're going to look at the human digestive system. So the main function of the human digestive system is to digest and absorb food. That is the process. That is kind of the principle behind what its job is. So when we're thinking about the digestive system, we need to consider a few different organs and tissues that are involved. So thinking about the journey of food through the digestive system, it starts off in the mouth, food is chewed to break it down, and amylase enzyme gets added at that point to start breaking down the starch into sugar. Food then passes down the esophagus into the stomach, where you've got the stomach acid, and you've also got protease enzymes that start to break down the proteins. Then into the small intestine, where various other enzymes get added, and the bile. And then the large intestine, then obviously exiting the body after that. Okay, so that's kind of the order of food travelling through the digestive system and some key organs that we need to know about. Additionally to that, we've got the liver, which is its main job is to produce bile, which we'll talk about in a second. Then we've got the gallbladder, which stores the bile that's produced in the liver, and we've got the pancreas, which has an important role in producing enzymes. And additionally, this links into paper 2, it also produces insulin to control blood sugar levels. So thinking about enzymes then, they are biological catalysts, they're proteins themselves as well, and the way we describe how they work is the lock and key theory. So what this is kind of about is the fact that we've got enzymes with a specific shaped active site that is specific to bind a certain type of substrate. So for instance, this enzyme here could be amylase enzyme, in which case the substrate that is the correct shape to fit that active site would be starch. Okay, so that's the lock and key theory, just like obviously a key is specific to only one lock on a door. With all enzymes, they have specific optimum pH and optimum temperatures, and extreme pH or temperature can denature the enzymes, and what that will actually do is it will change the shape of the active site. Therefore, if this enzyme here is supposed to be a denatured one of this one here, the starch substrate that we were talking about earlier wouldn't be able to bind, because obviously you can see it's not the correct shape anymore on the active site. So that's worth bearing in mind. They have optimums and they will vary depending on which enzyme it is, but extreme pH or extreme temperature above the optimum temperature will denature the enzyme and change the active site shape. Looking at our key examples of enzymes then, what we've got here is kind of a table that breaks it down. So 3 different types of enzyme: amylase, which is a type of carbohydrase enzyme, protease and lipase. Amylase comes from the salivary glands and the pancreas primarily. It breaks down starch into sugars such as glucose. Protease enzyme, produced in the stomach, pancreas and small intestine, breaks down protein substrate into amino acids. Lipase enzyme, from the pancreas and small intestine, breaks down the lipids, aka fats, into fatty acids and glycerol. Okay, so this is definitely worth learning off by heart. We need to know the enzyme name, the substrate name, where they're produced, and also that kind of smaller soluble product that is formed. Moving on then to the role of bile. So as we said above, it's produced in the liver, stored in the gallbladder. Bile has two really important jobs. The first one is that it emulsifies fats, so it takes large fat droplets and breaks them down into smaller ones to increase their surface area. So obviously larger surface area, they can then get broken down much more rapidly into fatty acids and glycerol by the lipase enzymes. So emulsifies fats to increase the surface area. Also, bile is alkaline, so it neutralises the stomach acid. Now, the reason this is particularly important is it's so that it creates the correct pH environment for the lipase enzymes to be able to work, and other enzymes, and break down the substrates that they're breaking down in the small intestine. Okay, so neutralising stomach acid. With regards to the enzymes required practicals, this is the one where you're looking at the effect of pH on the rate of reaction of amylase enzyme, so kind of looking at how quickly amylase can break down starch depending on the pH you put it in. The independent variable, the one that you change, is going to be the pH. The dependent one, the one that you're measuring, is the time taken for that starch to be broken down. And the control variables, there will be many, but the key ones are things like the temperature, because obviously if you're investigating pH, you need to keep the temperature constant, concentration and volume of the various solutions. So concentration and volume of your starch, amylase, and the same really with regard to kind of the volume of iodine and things like that you use, and the pH buffer. Okay, moving on to another big practical then that we had to do as part of Unit 2, food tests. So with the food tests, if the sample that you're starting with is not already kind of a liquid sample, you need to prepare that additionally prior to carrying out the tests. So to do this, what you would want to do is grind the food sample up with a pestle or mortar, add distilled water, stir it and then filter it so that you end up with kind of a liquid solution of the food that you're trying to test. When you're testing for the presence of sugars, you use Benedict's reagent, which is a blue coloured reagent. Add it to your food sample. Then you need to place it in a water bath for 2 to 3 minutes. If a positive result is observed, you will see a brick red colour, which will tell you that there is sugar present in that food sample. If you don't observe a positive result, it could be that the Benedict's kind of is still a blue colour, providing your food isn't also coloured itself, and obviously that would change the colour of the Benedict's as you mix them together. Okay, but for a positive result, brick red. Testing for starch then, you add iodine reagent, which is orange and brown in colour. If a positive result is found, you will see a nice blue-black colour, which will tell you that there's starch in that food. If not, you potentially will still be observing an orange and brown iodine colour. Testing for proteins then, Biuret reagent, pale blue colour on its own. Add that to your food sample. If a positive result is observed, you will see a purple colour. And then your final food group, your lipids, your fats. 2 methods for this one. So Sudan 3 stain, which is red on its own. If you add that to lipids and fats and it's a positive result they're present, you'll get a red oil layer forming. Additionally, you can do a test where you add ethanol and water to your food sample. If fats are present, you'll get a milky white solution forming. So that's your food tests, kind of summarised on that table there. Moving on then to look at the heart, it's another big section of this topic, so we're looking at kind of the heart and the blood now really. We'll start with, first of all, the 3 types of blood vessel. Okay, so the arteries, veins and capillaries. The key differences between them with regard to their structure and function are as follows. Arteries, really thick wall made of muscle and elastic fibres. They've got a smaller lumen than veins. And the reason this is because they need to withstand the high pressure of blood flowing through them. Veins, on the other hand, have slightly thinner walls, they've got a larger lumen, and they've also got valves to prevent backflow of blood. And the reason they can have kind of the thinner walls, larger lumen, is because that blood is under a lower pressure. The final blood vessel then, the capillaries, they're really narrow, they're the tiniest blood vessel of all. They're very thin walled, they're actually only 1 cell thick, and the reason this is so that diffusion across the capillary wall can happen really rapidly. With regard to the structure of the heart then, this is one that people often find quite tricky. So when it comes to drawing or labelling a heart structure, the first key thing to bear in mind is the fact that your rights and lefts are on the opposite side to what you'd think. Okay, so you can see we've got the right atrium and the right ventricle here, which obviously are on the left side of the paper, and then on the right hand side of the paper we've got the left atrium and the left ventricle. So you must learn the 4 chambers here, and also just bear in mind the rights and lefts are the opposite way round to what you would think. So blood from the body, when it's returning back to the heart for the first time, it's going to enter through the major vein called the vena cava, into the right atrium, through to the right ventricle, and then it's going to go out via the pulmonary artery to the lungs, where gas exchange is going to take place. So carbon dioxide will be dropped off, oxygen will be picked up. It's then going to come back into the heart via the pulmonary vein, into the left atrium, into the left ventricle, and then it's going to be forced out via the major artery called the aorta, where it's then going to go to the rest of the body to provide the body with oxygen, okay, the oxygen that cells need obviously for respiration. So those 4 blood vessels are super important. The 4 chambers you also need to be able to identify and label. Additionally, just to add to this, we've also got a little section of cells over here in the right atrium that are really important. Those are called the pacemaker cells because they control the natural resting heart rate. So they kind of make sure that the heart is contracting as it should be to keep this flow of blood through the heart happening. Just really quickly then, thinking about the blood itself, it's made up primarily of red blood cells, white blood cells and platelets, and those are suspended in a liquid called plasma. Okay, so those are the 4 components of the blood that we need to know. Red blood cells carrying oxygen around the body, white blood cells are really obviously important for the immune response, and platelets help the blood to clot. Looking at the lungs then next, which are the site of gas exchange, we need to know the fact that when we're thinking about the passage of air into the body, it starts off with air entering through the mouth and the nose. That air then goes into the trachea, into the bronchi, into the bronchioles then, which are kind of a smaller group of a little network of tubes, and then also it goes into the alveoli. At that point, what will happen is oxygen will pass over the alveoli wall into the blood that's contained within the capillaries, and carbon dioxide, which is your waste product from respiration, will pass out back into the alveoli, and then that will be exhaled. Okay, so air is passing in at the alveoli, oxygen will diffuse across the wall into the capillaries and bind to the red blood cells, and then obviously carbon dioxide will diffuse out as a waste gas. 2 more sections to cover. So looking then next at coronary heart disease, so CHD, what that is basically a condition where fatty material will build up in the coronary arteries, narrowing them and reducing blood flow. So with the coronary arteries, they are the arteries that surround the heart muscle itself and supply that muscle with oxygen and with glucose that it needs to respire, to produce energy so that the heart can continue to contract. So CHD, blockage of the coronary arteries. 3 main types of treatment: stents, which actually physically hold those coronary arteries open to allow the blood to continue to flow through past the blockage; statins, which help to reduce cholesterol and reduce the likeliness of a fatty build-up; and transplant as well. Okay, so if the heart has actually failed as a result, heart transplant might be the only option. Risk factors for CHD: high fat diet, lack of exercise and smoking, among others. Looking at cancer then next. Cancer can be defined as the uncontrolled growth and division of cells. If it's benign, it's a type of cancer that will not spread. If it's malignant, it can spread and it can form secondary tumours. Very final part then for this one, we've got the plants. So we need to know the structure of the plant leaf, and the following things: the xylem, the phloem, the root hair cells, the stomata, transpiration and